Welcome to Genetics Part 1, Mendel and the Gene. Our traits make us who we are. The expression of your genetic code determines how you look and how you function. In this unit, we will review simple genetic expression as well as look at more complex patterns of inheritance. As we begin, I want you to think back to earlier biology courses you've taken, and what do you remember about Mendel and his genetic studies? What are some of the genetic terms you remember? Take a moment and just make a list of the terms that you remember or that you think of when you think of Mendel and Mendelian genetics. Now I'd like you to pair up with someone near you and describe what you remember about each of the terms listed on the screen. We'll come back to this list at a later time, but pause for now and talk to someone near you about each of these. See what you can remember about these specific genetic traits. An organism who has two identical alleles for a character is said to be homozygous for the gene controlling that character. An organism that has two different alleles for a gene is said to be heterozygous for that gene that is controlling the character. Unlike homozygotes, heterozygotes are not true breeding. So because of the different effects of dominant and recessive alleles, an organism's traits do not always reveal the genetic composition. Therefore, we distinguish between an organism's phenotype or physical appearance and its genotype or genetic makeup. In the example of flower color in pea plants, big P, and big P, little p plants have the same phenotype, which is purple, but they have different genotypes. Here you see the P generation, where purple flowers were mated with white. Both true breeding parents, being homozygous, produced F1 hybrids that appeared purple. So you can see the phenotype, but the genotype is not apparent, although we would know in this case that it is big P, little p. When purple flowered plants from the F1 generation crossed, we get a very predictable 3-to-1 ratio of purple to white flowers in the plants found in this F2 generation. So Mendel chose to track only those characters that occurred in two distinct alternative forms. He also used varieties that were true breeding, plants that produce the same variety when they self-pollinate. Mendel was unaware that traits were carried on our genes. He proposed that the units of inheritance were discrete particles in what he called the particulate theory. For example, the gene for flower color in pea plants exists in two versions, one for purple flowers and the other for white flowers. These alternative versions of genes are now called alleles. Each gene resides on a specific locus on a specific chromosome. For each character, an organism inherits two alleles, one from each parent. Mendel made this deduction without knowing about the role of chromosomes. The two alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation. Alternatively, the two alleles at a locus may differ, as in the F1 hybrids. If the two alleles at a locus differ, then one, the dominant allele, determines the organism's appearance, and the other, the recessive allele, has no noticeable effect on the appearance. In the flower color example, the F1 plants had purple flowers because the allele for this trait is dominant. We'll use what Mendel concluded, along with contributions from other geneticists, to predict patterns of inheritance. By the time we are done, we will have practiced calculating the inheritance of traits in monohybrid crosses, dihybrid crosses, and those that are sex-linked, as well as those that are autosomal linked. Let's begin by looking at some of the fundamentals of genetics as presented by Mendel. Let's start with the law of dominance. When Mendel crossed F1 pea pods, he noted yellow pods in the F2 generation. The green allele had masked the expression of the yellow pod color in the F1. The yellow pod color could be expressed in the F2 generation when the recessive allele from one parent combined with the recessive allele from the other parent. Dominant alleles are those that mask the appearance of other alleles. There are some human traits that are thought to be controlled by single alleles expressing dominant. Some examples are given here on the screen. Widow's peak, right or left-handedness, polydactyly, even hair on your fingers. Those are traits found in humans that we think, at this time at least, are inherited by simple dominance. The allele for this unusual trait is dominant to the allele for the more common trait of 5 digits per appendage. If you look closely at this hand, you'll see there's 5 fingers and a thumb. That's a little different. But just because that trait is dominant doesn't mean it's going to have the highest frequency. It just means that if that allele is present, it is going to mask the recessive allele when the recessive allele is present. Why don't you take a moment, think about that, and apply it to this question. In humans, alleles for dark hair are genetically dominant, while alleles for light hair are recessive. Which of the following statements are most likely to be correct? Have you chosen your answer? I hope you selected B. Some people think, incorrectly, that the genetically dominant allele will be most frequent and that a dominant allele is going to increase in a population over time. The Hardy-Weinberg law shows that this is not the case. Dominance and frequency are not related. Allele frequency of dominant alleles could be low, like dark hair in Northern Europe, or dark color in Southern Europe. Answer B, in this case, is the only answer that reflects this pattern. Mendel's law of segregation can be explained more readily now that we understand the concept of chromosomal inheritance. During the process of meiosis, homologous chromosomes will separate into different gametes. This separation creates gametes that show variation. The law of segregation explains that the 2 alleles for an inheritable character separate or segregate during gamete formation and end up in different gametes. Thus, an egg or a sperm gets only one of the 2 alleles that are present in the organism. Mendel's segregation model accounts for the 3-to-1 ratio he observed in the F2 generation of his numerous crosses. The possible combination of sperm and egg can be shown using a Punnett square, a diagram for predicting the results of genetic crosses between individuals of known genetic makeup. A capital letter represents a dominant allele, and a lowercase letter represents the recessive allele. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. Segregation leads to variation in gametes, ultimately giving variation then to the offspring. Variation is one of the primary advantages of sexual reproduction. It is this variation that provides adaptability for the offspring. How can we tell the genotype of an individual with a dominant phenotype? Such an individual could be either homozygous dominant or heterozygous. The answer is to carry out a test cross, breeding the mystery individual with a homozygous recessive individual. If any offspring displayed the recessive phenotype, the mystery parent must then be heterozygous. Mendel identified his second law of inheritance by following 2 characters at the same time. Crossing 2 true breeding parents differing in 2 characters produces dihybrids in the F1 generation, heterozygous for both characters. A dihybrid cross, a cross between the F1 dihybrids, can determine whether 2 characters are transmitted to the offspring as a package or if they're transmitted independently. Using this dihybrid cross, Mendel developed the law of independent assortment. The law of independent assortment states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. Strictly speaking, this law applies only to genes on different non-homologous chromosomes or those far apart on the same chromosome. Genes located near each other on the same chromosome tended to be inherited together. In order to solidify your ability to explain the laws of segregation and independent assortment, take a moment to stop, pause, and describe how these laws would apply as sperm cells are developed in a man whose genotype is big R, big S, little s. And then on a piece of paper, draw an image that can support your explanation. Here's a sample image that you may have prepared. The alleles for each trait separate from one another during segregation and will end up in different gametes. Notice that in this diagram, the big R is never in the same cell with the little r. The big S is never in the same cell with the little s. Independent assortment refers to the manner in which traits will sort without regard to the other trait, provided they are carried on different chromosomes. The 4 cells are drawn to show the various combinations that can occur. Find a scratch piece of paper, something to write with, and let's consider this question. Imagine crossing a pea heterozygous at the loci for flower color, white versus purple, and seed color, yellow versus green, with a second pea plant homozygous for flower color, white, and seed color, yellow. What types of gametes will the first pea plant produce? Have you finished your calculations? This question requires that you figure out gamete types. This step is often rushed past. We need to realize that the gametes are haploid and that each gamete contains one and only one allele for each of the traits being studied. The presence of a second P in the question stem here is a distractor. Answer A is wrong because it shows gametes with 2 alleles for flower color and no allele for seed color. Answer B is wrong because it shows only 2 possible gamete types. Answer C is the correct answer because it shows all 4 possible gamete types. Answer D is wrong because it shows gametes that are diploid for one trait and containing an allele for only one locus. So this has been a review of some of the more common terms that we use in genetics. This should serve as a foundation for you as we move into part 2, in which we will predict the frequency of occurrence of specific offspring types.